Alpha-1 antitrypsin deficiency, or Alpha-1 as we say for short, has been described for just over 50 years now, and the progress that has come in this time is just phenomenal. So first described in 1963, and Alpha-1 is a single gene disorder, meaning that the way your body makes this Alpha-1 antitrypsin protein is determined specifically by a single gene. And it's a gene that you receive one copy of from your mom and one copy of from your dad. So everybody has a total of 2 Alpha-1 genes that encode your Alpha-1 protein production. Disorders that occur based on a single gene are said to be Mendelian. And here we have a picture of Gregor Mendel up there. He's termed the father of modern genetics. He lived in the 1800s and was an Austrian friar and scientist, and is known for his crosses with pea plants, among other things. But it was his work that really gave way to the idea that there were units of heredity or discrete factors that were transmitted from generation to generation. A reason that families share traits. Kids look like their parents and sometimes share health conditions with their parents. So out of his work, single genes were identified and issues of dominance and recessive traits were identified. So in some cases, maybe brown eyes trumps blue eyes. If you have one brown gene and one blue gene, you're going to show up with brown eyes. When both genes play a role in what you manifest, that's something we call codominant. So again, some tricky words in the genetics segment. I'll be talking about codominant expression and also recessive inheritance as we look at risk to our children and downward generations based on our own Alpha-1 genes. We've talked about the Alpha-1 deficiency. What is Alpha-1 or Alpha-1 antitrypsin itself? We had a fantastic review this morning about the protein Alpha-1, and that's a really important concept, is that Alpha-1 is a protein. And it's mostly produced in the liver. Then it's supposed to leave the liver and circulate in our blood, where its job is to protect our lungs from damage by binding that neutrophil elastase molecule. The deficiency is a term for not enough circulating Alpha-1 antitrypsin, or not enough functional Alpha-1 antitrypsin to provide adequate protection for our lungs. And we know that in some cases of Alpha-1, it's actually an overload of the Alpha-1 molecule in the liver that raises risk for liver disease as well. But part of why knowing your genotype is so important is that not all Alpha-1 genes are created equal. So not all confer the same degree of risk for lung symptoms or the same degree of risk for liver symptoms. So we know what Alpha-1 is. What are genes? Genes are that unit of heredity, a discrete factor that is made up of DNA and serves as the biological instruction manual for how a protein's produced or how a protein's form. So genes are the blueprints or the recipe for making proteins, a vast array of proteins that are necessary for our body's growth, function, and development from conception all the way throughout life. People have about 20,000 genes total. They come on 46 chromosomes. Those 46 chromosomes are in 23 pairs. We get one copy of each from our mom, one copy of each from our dad. So hence we're getting 2 copies of each gene. And the genetic sequence of a gene determines whether it makes its protein normally or abnormally. So this picture shows if we were to crack open a cell, inside the cellular nucleus are our chromosomes, and those are large units of DNA. Each chromosome contains many, many genes. And if you unravel the DNA, there are specific subsets of DNA that make up each gene, which is encoding for a particular protein. And we saw those great diagrams of the Alpha-1 protein earlier. So if you have the normal DNA sequence, it's going to make the normal M Alpha-1 protein. But if you have an abnormal DNA sequence or an error in the instruction manual, you end up with a faulty product. The Alpha-1 gene is called Serotonin A1, and it's located on the long arm of the 14th chromosome. So if you actually made a picture of all the chromosomes inside of that cellular nucleus, we see something like this, where you have 2 copies in each of 23 pairs. They're lined up from the biggest as number 1 down to the smallest at 22, and then the last set is our sex chromosomes, determining whether we're males or females. So the 14th chromosome here is the culprit when we think about Alpha-1, and we know everybody has 2 copies of the gene. Now, genes for the most part contribute to our variation and why you don't look exactly the same as the person next to you. So you can have changes in your DNA that just make you unique. but still unique within the normal construct of how you're supposed to function. Different versions of the same gene are called alleles, kind of normal alleles, or you can have abnormal alleles. Abnormal alleles being those that cause an abnormal protein or lead to an increased risk or a disease. And abnormal alleles are called mutations. Your genotype is the combination of two alleles that you have. So if you're being asked your genotype, it's which two versions of the alpha-1 gene did you inherit? And we'll spend some time talking about each of the common versions of the alpha-1 gene. And that's what determines your personal risk. So what level of risk are you born with based on your alpha-1 genes? And it determines your risk to relatives, since those genes do run in families. Each gene has a usual spelling or sequence for its usual function. And the genetic alphabet is made up of A's, T's, C's, and G's. And it's those letters that say which string of the pearl should be put on next. Is it going to be the right one, or are we stringing something on there that ends up in the protein being misfolded and not working correctly? So it's important to have a normal spelled gene for normal function, kind of like if you were reading a recipe and they meant to tell you to put in sugar to bake a cake, but someone wrote salt instead. If you follow that instruction, you're still going to end up with a cake, but it's not going to please your guests so well. If you put the wrong letter in your genetic sequence, you end up with a protein that doesn't do its job so well. Another example of genetic mutation would be in the English language. If you're typing a sentence and you put a typo in, you can change the meaning of your sentence. So just a single letter can make a difference. If we tried to say the cow jumped over the moon, but instead put a T, say the cot jumped over the moon, well, it's something, but it's not a very good something. Likewise, you could be missing a whole word, and you see when there are many, many letters, there are many opportunities for error. So not all errors are the same. Alpha-1 alleles, so different versions of the alpha-1 gene. There are a few common ones that we think of, and we know now that M is the normal sequence in the normal protein. And about 92% of Caucasians, so most people, have two copies of the normal M, meaning their genotype is MM. The S and the Z allele are the most common abnormal alleles, and each of these are caused by a single base pair substitution in the genetic code. So just one letter gone awry that causes the wrong pearl to go on the string, and the protein gets misfolded or doesn't work like it should as a result. The S and the Z make up about 96% of all abnormal alpha-1 alleles. So the vast majority of people who have alpha-1 have it due to a ZZ or an SZ genotype. But there are some other rare alleles and null alleles, and we'll talk just a little bit about those as we get a little bit deeper into the genetics. Codominant means that one version of the gene doesn't trump the other. So if, say, you have an M and a Z, your M doesn't trump the Z and produce only M, but rather your M and your Z are both active and both produce protein. So in a person with two different letters, you're going to be able to detect both versions of the protein. An SZ individual, for example, would have both S protein detectable and Z protein detectable. An MZ person would have M and Z protein detectable. But if you had both M's, you're going to have 100% M protein. If you had both Z's, you're going to have 100% Z protein. So that lends to intermediate states when it comes to alpha-1. It means that your carriers or your heterozygotes, people with two different alleles, are going to have an intermediate level of alpha-1 that's not as high as your normal MM, but it's not as low as your severely deficient ZZ either. So we have some gray zones when it comes to alpha-1. We also see that our proteins correspond to our genes. So you hear this word phenotype. Genotype, we said, is looking at the level of the DNA, so gene, genotype. Phenotype is what you can visualize if you do a protein test. It comes from the same roots as the word phenomenon, so you can visualize it. It's the product of the DNA. And if you look at the alpha-1 proteins from your blood on a gel in the lab, it's referring to the protein that's present on the migration of the protein across the gel. So the M protein actually got its name because it moves at a medium speed, and the others were named with retrospect to that. Again, with the cake analogy, if you look at a chocolate cake, you figure it came from a chocolate cake recipe. If you look at a chocolate cake recipe, you figure it's going to make a chocolate cake. So let's say you look at somebody's results and you see a phenotype Z. You've seen the Z, you haven't seen anything else. You can infer that that person has Z protein present in their blood due to a Z instruction manual in the DNA. And if you do a DNA test and you find a Z mutation, and say, well, that person's going to produce Z protein as a result. So almost all the time, your phenotype test results, which is a protein type test, and your genotype test results, which is a DNA-based test, are going to give you the same 2 letters. And everybody has 2 letters when it comes to alpha-1, representing both versions of your alpha-1 gene. The exceptions would be if you're on augmentation therapy or have had a liver transplant, then you may have alpha-1 protein detectable in your blood that wasn't made based on your own genetic code. So there are some caveats that we keep you aware of. But hopefully that's clear that your phenotype and your genotype are concordant most of the time. Yes?
So when you go to your doctor, what do you do the test kit? Which way are they doing it?
So the question is about the method of testing if you go to your doctor or order the test kit through the alpha-coded testing study, what testing is being performed. And that does depend on which lab the testing is being sent to. With the alpha-coded testing study, it's a genotyping test, so looking at the DNA for common mutations and also measuring the quantity of alpha-1 protein in your blood. Oftentimes, several methods are employed to make sure that detection rates are good and you get concordant results on several different methods. But there are labs that perform phenotyping as a first-tier test and several acceptable paradigms. So it's something that sometimes I'll ask you if there's any reason to suspect that you might benefit from another test. Most of the time, whatever test your doctor orders for you is one that's been vetted and is medically
Well, that's
sound.
what I was going to ask. Phenotype is accurate enough that you don't need to request a certain one.
So measuring the quantity of alpha-1 in your blood, so an alpha-1 level or an immunoassay supports a genotyping test or a phenotyping test, because the tests that look for just your most common mutations, like your S and your Z and sometimes a couple others, can't detect all rare and null alleles. But if your alpha-1 level was measured and is still really low, that would be a tip-off to maybe order a gene sequencing test, which is a more advanced method. Same goes with phenotyping. Ordering that level in combination with your phenotype can just serve as a check that everything's clear and correct in your personal case. A level alone cannot tell you your genotype or your phenotype. So that's the one that I'm less comfortable with as the genetics person, because I can't tell you what it means for your family. We can say whether you're severely deficient or not, but you can't separate your carriers from your MMs or say if it's an S or a Z at play. So it is a good question to ask your doctor, and if you're able to, get a copy of your own results, especially anything genetic. You usually don't need to repeat the tests in your lifetime, but people get very confused in what the results say, and sometimes the lab methods do matter. So if you have it in a file somewhere, save it to your hard drive or whatever you're comfortable with so you don't lose it. It can help in looking back and making sure that everything was proper. Yes?
So I'll emphasize the importance of knowing, emphasize the importance of knowing your information. So when folks come to see us and they're looking for an opinion and clarity, it's really important that we have the primary test results. You may remember it, but it's even better if you have it on a piece of paper from your doctor's office or from the University of Florida. If you've gone through the ACT, the Alpha-1 coded testing, you'll get in the mail a piece of paper that says your level is this, either in milligrams per deciliter or in micromolar, or both in that instance, and you'll get the letter combination, which is, of course, as Kim has so nicely explained, the genotype. It's really important to have the primary information in front of us, because if we're guessing, there's so many opportunities to get it wrong, either because you don't remember or we're trying to infer. So a great help for those of you going to see your doctors with this would be to get the information together with a hard copy of the information in your hand, ideally sent. When I see folks, many of you with whom I've had a chance to see will know that my office will try to reach out to you to get those records in advance of your visit so that I have time to look at the x-rays and the lab tests. And so when you come, I can pay attention to you as opposed to trying to go through a telephone book of records in the moment. So it's really important to know that information and to get it in advance. If you're seeking an opinion from Charlie Strange or Mark Brantley or Sandy Sandhouse or Jim Stocks or Ed Silverman or me or Umar Hadapolu, if you're really going to that much trouble and expense to come and see us, it's really important to know that information at the level Kim is describing.
Thank you. Certainly agree. And if anyone has questions about their own method of testing, it's part of what I'm here for, so give me a call or feel free to grab me on the next break. It's always good to have those records and have an understanding so you can clearly incorporate that information moving forward. So our genotype categories give us those with severe deficiency or Alpha-1 diagnosis on the laboratory side when we see two abnormal genes, either your ZZ or your SZ combination, and that results in a corresponding low level because those are the genes that aren't making the Alpha-1 protein normally. Now, our carriers are people who have one normal M gene and one of something that's not an M. So our carriers are usually MS or MZ, but since there are other gene abnormalities too, it would be M and something that's not an M. And carriers can also be said to have a mild deficiency with that intermediate level. And we know Alpha-1 detection is ever so important because it does influence your health decisions and management and your family testing. We'll quickly review the Z allele. We learned a lot about the Z this morning and how the misfolding of the protein based on the Z mutation causes that Z alpha-1 protein to be sticky and glob up and form polymers that get trapped in the liver. So you end up with an alpha-1 overload in the liver cells, raising the risk for liver damage, and a resulting serum deficiency because most of it isn't getting out to circulate in your blood and protect your lungs. So people with two Zs are our classic alpha-1 and have a level of less than 11 micromole or less than 50 milligrams per deciliter. And those numbers, again, will come from the level assessed on your Alpha-1 blood test. Blood testing is necessary to diagnose Alpha-1 and tell you what strain of Alpha-1 you have, if you will. And about 2 to 3% of Caucasians are carriers for the Z allele, so having the MZ genotype. And carrier detection can be really important when there's Alpha-1 in a family if people are thinking about having children, because we know when both parents are carriers or one parent's affected and the other's a carrier, that's where there's a risk to the next generation to inherit the severe Alpha-1. Further, MZ carriers should really strongly avoid cigarette smoke and additional insults because given that mild deficient level or intermediate level, the intrinsic protection to the lungs is just not quite the same on average as your normal MM individual. Now the S allele is an intermediate allele. It is more effective than the Z, and it doesn't polymerize in the liver in the same way as the Z, but it's not as good as a normal M either. About 5% of Caucasians carry the S allele, and an S allele when combined with another S allele will give you about 60% of the normal Alpha-1, which in general, in the absence of other major risk factors, is adequate to protect your lungs. And the S isn't associated with that liver risk. But an S, if combined with the Z, does pose a moderately increased risk and constitute Alpha-1. So S's can be important to be aware of as well. I'll just quickly mention some other rare alleles, and if you have these in your family, give me a call. We're all still learning a lot about rare alleles, and advances in genetic testing and in Alpha-1 have really made this possible. The alpha-coded testing study just recently added detection of the F and the I allele, and I know they're on some other tests as well. The F allele is a tricky one because it produces an Alpha-1 protein that circulates at a normal level but doesn't work right. So that can be really confusing, and you need the right laboratory test to detect it. Fortunately, it's pretty rare. And most of the time, it's not something that you need to worry about, but as our testing gets better and we really want to make sure we're not missing people, we have added these two to that alpha-coded testing study, which is the free and confidential testing program supported by the foundation and through the Medical University of South Carolina. So if anybody does have questions about that, I can answer those as well. It's a great way to get your family members tested if they're not going to your doctor's appointments with you. Now, null alleles are a family of alleles. Null's an umbrella term and not a specific gene mutation itself, but it's a family of gene mutations that instead of producing an abnormal protein product, just shuts down the function of that alpha-1 gene and causes no alpha-1 protein to be produced. Nulls are important because we have to be thinking about them on the scientific and medical side when we're making sure you had a test capable of detecting nulls if, say, you have a family history of a null allele, but also when it comes to personal health, because null alleles produce nothing, there's no liver risk associated, but you have the most marked serum deficiency, so lung risks are going to be as high as they go on the inherited side of alpha-1. We already touched on finding out your genotype a good bit. Here are some of the technologies applied on the back end. So genotyping is actually looking at the DNA for common mutations like the S and the Z, and perhaps the F and the I. Phenotyping is that protein typing test or visualizing the protein product of the gene. So you can make inference between those two pretty certainly in most cases. The immunoassay or the alpha-1 level is the quantity of alpha-1 in your blood, and that can support those. And if there's anything tricky about those first tiers of testing, alpha-1 gene sequencing is actually taking the alpha-1 gene and going through letter by letter and detecting any changes in the spelling of the gene that could cause problems. So that's a more advanced method, but it's becoming more and more feasible and applied more often as things move forward. This slide just reviews some of our common genotypes from MM down to our ZZs and our nulls, along with our risk levels. So our MMs aren't off the hook when it comes to COPD or liver disease because anybody can develop COPD or liver disease. But related to alpha-1, if you're MM, you have no increased inherited risk. When we get down to our ZZs, we see high risk for lung disease, significantly increased risk for liver disease over the general population just based on those genes. Null, high risk for lung disease, but not for liver disease. So you see that your genotype allows us to risk stratify for you, but the genes really are not a fate. Alpha-1, when we look at the genes, is a laboratory diagnosis. It doesn't tell you that you are going to get sick, how you are going to get sick, what your progression is going to be like. So the genes tell some of the story and let us help you from there, but they don't tell it all. I like this genetic threshold model saying that there are other forces at work to influence who actually does present clinically with COPD. That MZ people have their genetic jar sort of half full, but it may take more environmental insults for them to manifest the disorder, whereas ZZ people are born with a large genetic burden toward manifesting the clinical signs of alpha-1. And here's also just a slide depicting the multifactorial presentation of your clinical phenotype, which is a large scale, what you can see or how you present to your doctors or how you feel, where there are probably lots of environmental factors and probably other genetic susceptibilities or protective factors influencing who manifests severe alpha-1 versus lives into their 70s or beyond without having problems related to it. So we do know alpha-1 is a family matter. When it comes to the reproductive risk, we say that that is recessive, and that means that the risk for severe disease or two abnormal genes occurs when both parents have an abnormal gene to pass on, most commonly in the event that both parents are carriers. So presumably unaffected carriers since most MZs don't manifest disease. And when both parents are carriers with one normal and one abnormal gene, there's a 25% risk to each child to inherit the severe genotype or both abnormal gene copies. So everybody has two copies of the alpha-1 gene. You pass on one or the other each time you have a child, 50-50 chance which one. So when you factor that in for mom and for dad, you look at these reproductive outcome possibilities. It means that full siblings of anybody who's been diagnosed with alpha-1 are at risk to also have alpha-1 because they shared the same mother and father. So alpha-1 testing for siblings is really, really important because early detection and care leads to better outcomes. And anytime a carrier is found, all their first-degree relatives have a 50% chance to be a carrier as well. So if we identify just an MZ carrier in the family, their parents, siblings, and children have a 50% risk to carry that same abnormal gene. And when one parent is the alpha, of course, they're going to pass on one of the abnormal genes to the child, and the child gets the other alpha-1 gene from the other parent. So we always hope that the other parent's normal MM, that's most likely, which would give us a 100% chance of children being MZ carriers. But since there's that background carrier risk, carrier testing is the only way to tell for sure. So offering carrier testing to the partner of any known alpha or alpha-1 carrier is important. And here we have a Punnett square, kind of comes out of Mendelian genetics as well. And you write the parental genotypes or alleles that each parent has across the axes, so one across the top and one down the side. And just pulling in those letters into your block shows you your genotype possibilities and probabilities for the offspring. So that's a really foolproof way to see what the chances are to the next generation. Put this up here, alphabet soup needs more punctuation. I know sometimes the genetic code and all the alpha-1 alleles can be a lot to wade through, so my job is to be here to help you understand that. I'll just very quickly tell you a little bit about the genetic counseling program. That's me at my desk in South Carolina. And you can reach me at this toll-free number. It's a free program generously supported by the Alpha-1 Foundation to make sure that you all have access to good information when you're considering testing or have received your test results or wondering what's next in your Alpha-1 diagnosis or journey. And common reasons that people call are whether and how to be tested, what results mean for their own personal risk level, perhaps recommendations of the closest clinical resource center or Alpha Doc to them, how to get involved in a support group or in research opportunities, what do results mean for their family members and how can their kids or their siblings be tested, really helping them on the road from wherever you start to wherever you need to go. So there's no question that I won't do my very best to help you with, and I think a lot of the time I'm successful in doing so and always happy to hear what I can do for you. Do give me a call. I'm out of time. The last thing I have is a case example, which I could skip, but if we have like 2 minutes, I'll run through it. So oftentimes when you do call me as a genetics person, I'll sketch out a little pedigree or diagram of your family tree. It shows our generations of the family, and we have squares and circles connected to each other by lines. Our squares represent men and our circles represent females. A line between the shapes connecting them is a mating pair, and then you see the next generation coming off of that. So let's say this man, Jeff, calls me up on the phone, and he tells me he's a 66-year-old man, and he's had COPD for 5 years. He never smoked, so he couldn't figure out why he had COPD, and he was finally tested for Alpha-1. Comes back with a ZZ, severely deficient diagnosis. So he says he's relieved that he finally has a diagnosis, but he's kind of frustrated that it's been 5 years into his journey and nobody tested him or even told him what Alpha-1 was before then. So we'll have a good conversation about what Alpha-1 is, and of course many of you can unfortunately relate to that sort of diagnostic journey. We get him to go to an Alpha Doc. He's going to begin augmentation therapy, get involved in research, get involved in a support group, and all the wonderful things that the community offers to alphas that many of you know about. But he has all these other relatives connected to him in his family tree as well. We see that this man here is a brother to Jeff, and Jeff's diagnosis informs us that both of his parents must have been Alpha-1 carriers in order for him to be ZZ. So we know that his brother is at risk for Alpha-1 as well and should be tested. So how are we going to handle that? And then Jeff wonders if his mom's death could have been Alpha-1 related. We know now that she must be a carrier, and she died of emphysema at 68, and he wonders if she could have lived longer if the family had known about Alpha-1. Further, he has 2 daughters who have definitely gotten a Z from him, and hopefully are just MZ, but only testing would say for sure. And in this scenario, one of them's pregnant, so we might want to test her partner to see if there's a risk to that child. and the other one's a smoker, so smoking cessation counseling for them. And not to forget his spouse. As you know, oftentimes our spouses become our Alpha-1 champion and cheerleader and support person. And he says she doesn't know a whole lot about Alpha-1, so we need to get her in the loop as well and support for the support person. So this is a lot of real-world issues that we deal with every day in the Alpha-1 genetic counseling program. And I'm always happy to talk through your personal situations, answer your genetics questions, but also how we can get your families plugged in, educated, informed, involved as well.